Okay, we will go ahead and get started. Welcome, everyone, to our next Black in Neuro seminar series. My name is Brielle Ferguson, and I'm a postdoctoral fellow at Stanford, and I'm a co-founder and director of programs for Black in Neuro. Before we start, I want to give people a heads up that if you look in the bottom right corner of your screen, there should be an option to access the live transcript if you would like. Additionally, if you want to share any tidbits that you learned today on social media, please do so using the hashtag #BIN seminar series. Next, I would like to acknowledge that I am joining you today from Ramaytush, Ohlone, and Muwekma lands. We're all joining this event from places around the world, so I invite you to take a moment to reflect on the lands that you are joining from, as well as your relationships to those lands and the indigenous people who have and continue to inhabit them. If you happen to have missed any of our seminars, I highly recommend that you go check them all out. They're all on our YouTube channel, and we'll have someone drop that link in the chat. As I say in those seminars, and our other organizers on the seminar organizing committee have said, this series is just so important to us for so many reasons. For Black scientists, we're not used to seeing ourselves represented in our cohorts at our institutions, and especially not in faculty and leadership roles. So that's why introducing this next speaker is so special to me. Dr. Corey Harwell is a native of Louisville, Kentucky, and obtained his Bachelor's of Science in Chemistry from Tennessee State University, and then his PhD in neuroscience from MIT. After receiving his PhD, Harwell went on to complete a postdoctoral fellowship in Arnold Kreigstein's lab at the University of California, San Francisco. During his postdoc, he identified a role for sonic hedgehog and its receptor VOC1 in specific facets of neural development. Following his postdoctoral work in 2013, Harwell became an assistant professor at Harvard Medical School in the Department of Neurobiology, where he continued to make important discoveries toward better understanding neural development. Just this year, during a whole pandemic, Dr. Harwell moved his lab to UCSF, which is super exciting, where I know he'll continue to push the field forward, and I just can't wait to see what he does next. I personally first met Corey back in 2016, where he taught at a Cold Spring Harbor course, and I remember just being so blown away that he existed, because at that point I'd met 0 Black faculty. And I just was so excited that I was going to have the opportunity to be able to interact with him and just like pick his brain and hear what his experience had been like. It was a really big turning point for me in terms of believing that I could actually make it in this field. And I hope that it will do the same for some of our audience members. Research in the Harwell lab has been focused on understanding the genetic and epigenetic mechanisms that guide progenitor cells and how those cells in the brain choose their mature neural fates. Today, he'll talk about development and diversity of neural cell types in the septum. So without further ado, I will pass it off to Dr. Harwell. Thank you so much.
Thank you, Brielle, for that really, really generous introduction. And I do have to admit that when we were hearing about you starting your postdoc and now looking, at some point in the near future for faculty positions, it does make me feel a little bit old to remember that back in 2016, you were but you were still a graduate student. So, and now kind of mid-senior level postdoc. So time really flies. And it's been, inspiring to see your career trajectory, and I can't wait to see what you go on to do. So I'm going to talk about some stuff that's kind of a mix of mostly new stuff from the lab that we're actually pretty excited about, and especially with our new environment at UCSF, where we think that we can take this work in some really cool directions. So we've recently become really interested in the structure in the ventral part of the basal forebrain called the septum. And I have to say that I've learned that it's not a very well-known structure, even for a lot of neuroscientists. But I think that there's a lot of kind of growing interest in its structure, given its role in regulating behaviors that I'll talk about in the background. But here's just a coronal section of an adult mouse brain that many of you are probably familiar. And the septum is just located actually in the medial parts of the ventral forebrain. and more rostral medial, to be more precise. And it's principally divided into these kind of medial and lateral nuclei. And the next question we always get is, what does the septum do? So what is its function? So it's thought to function as a link between the limbic system and these higher order cognition areas, including structures like the hippocampus, entorhinal cortex, prefrontal cortex. And it's thought that through these links, it controls innate behaviors related to motivation, anxiety, and aggression. And in humans, dysfunctions in the septum are thought to be implicated with psychotic spectrum disorders and anxiety and depressive disorders. Oops. If you look at the neurons that comprise the septum, they're primarily composed of GABAergic and cholinergic neurons. However, there are also a subset of glutamatergic neurons that are located also within the medial septum. And I think one of the reasons that we became really excited about the septum from the perspective of developmental biologists is that there's an incredible amount of diversity in the neuronal cell type. So this is just looking at the lateral septum, which we've focused on quite a bit. And you can just appreciate just from the morphologies, the extent of the diversity, but that diversity also extends to some of the molecular identities and their intrinsic electrophysiological properties. And one of the, I think, really big attractions for us as developmental biologists in looking at the septum is that it's compared to, I think, a lot of other forebrain structures, such as the cortex and the striatum, or even GABAergic interneurons, that the development and specification of neurons in the septum is relatively unstudied, or understudied, I should say. However, some of the kind of fundamental features of septum development have been studied and researched. So we know that, for instance, that the general patterning of the septum follows this, we should say, medial to lateral patterning, where the earliest born neurons typically populate the more medial structures in the medial septum. And then as neurogenesis progresses, more late-born neurons will populate these successively more lateral waves of neurons that will eventually populate the lateral septum. And there's also been, some key discoveries on the kind of molecular or the progenitor origins of neurons in the septum. So here's just a cartoon from a review we did a few years ago of an embryonic mouse brain. And the portions of the cartoon that are colored are the proliferative zones that give rise to all of the GABAergic cell types within the forebrain. So for instance, these zones in the embryonic mouse brain colored orange and red here for the CG and MGE give rise to forebrain interneurons, here colored in orange and red in the cartoon of the mature brain. And here in green is the LGE, which gives rise to the GABAergic projection neurons that will comprise the septum. And here in purple, finally, that's in this proliferative zone that's located just kind of ventral medial to the MGE, in fact, is this structure we call the POA and septum. And it's this structure that gives rise to the majority of those GABAergic and cholinergic neurons that make up the septum. And work from Nicoletta Casares' lab from several years ago identified these transcription factors that essentially identify the anatomical proliferative zone for the septum here. And it's these ZIC family transcription factors, here shown fate mapping with ZIP4 Cree. that gives rise to the septal cell type. So you can actually see the outline of the septal proliferative zone here in green using the SIG4-CRE reporter. But she also showed that in more caudal parts of the septum, there's this region that's labeled with the SIG4-CRE that's green, but these progenitors actually co-express another transcription factor called NKX 2.1. And we've gone on to acquire some of these tools from the Casares lab and other groups, and we've essentially been able to kind of confirm a lot of the fate mapping. For instance, that the SIG4-CRE does indeed give rise to the majority of the lateral septal neurons, shown here using a SIG4-CRE SUN1 GFP. But we've also done fate mapping using NKX 2.1 CRE to look at the production of those NKX 2.1 septal progenitors. And we initially noticed this really interesting pattern. So there's a large subset of neurons and glia that come from NKX 2.1 progenitors that populate the medial septum. But then there's this really interesting subset of neurons that are derived from NKX 2.1 expressing cells as well. Again, this is the lateral septal neurons in particular are coming from that region of progenitors that co-expresses Zik family transcription factors like Zik4. And this region, we've actually begun to differentiate and look at the specific output of those neurons that are coming from that Zik4 NKX 2.1 co-expressing region. So here we're using an intersectional genetic approach where we can take a Zik4 CRE expressing animal, an animal expressing another recombinase, in this case NKX 2.1 flip, and an intersectional reporter. And in this way, we can differentially label the cells that co-express NKX 2.1 and Zik4 CRE, which are in green here, from the cells that only express NKX 2.1 flip, which are the cells that are outlined in red. And we've come to refer to these septal neurons that are coming from this Zik NKX 2.1 co-expressing population of progenitors that's as the septal eminence. As Casares had showed, that it's this population that's located actually in the more caudal parts of the septum, where you find this co-expression of NKX and Zik4. And then you also notice that there's a subset of neurons within the medial septum that come from NKX 2.1 only expressing progenitors. And work from, again, from Nicoletta Casares, from John Rubenstein, several other groups, it showed that it's likely that these medial septal neurons are coming from this more caudal part of the proliferative zone that exclusively expresses NKX 2.1. Whereas for what we call either the septal eminence or the Zik-only region, which we refer to as the septum proper, gives rise to the majority of either the lateral septal neurons, and from the septal eminence, also the subset of cholinergic neurons that will go on to populate the medial septum. So one of the first questions that we had is that we were curious about, what are the molecular and functional features of these neurons that are coming from this caudal septal proliferative zone that co-expresses NKX 2.1. So we decided to utilize single nuclei sequencing to actually see what might be the specialized molecular properties of these cells in the septum that are coming from these NKX 2.1 expressing progenitors. So to do this, we took out the mature septum here, outlined at P30, and using this NKX 2.1 cross to this Cre-inducible Sun1 GFP, which is a GFP tagged to the nuclear membrane, so that we can sort out those nuclei from neurons that are coming from those NKX 2.1 progenitors and separate them from the other nuclei within the mature septum that are presumably primarily coming from those Zik expressing proliferative zones. And then we can do 10x sequencing and start to look at the molecular profile of the different cell types by using these graph plate-based clustering methods that I'm sure a number of you are familiar with. So this is just a UMAP of all of the cells that we've collected. I think at this point we've collected about, I think it's about 30,000 cells or nuclei or so from this experiment. And so what these UMAP plots can tell us is that each of the dots on the plot correspond to one of the nuclei, and the nuclei that have kind of transcriptional profiles that are more closely related will cluster together, and those clusters we take to be representative of different cell types. So for instance, if we look at a diagnostic marker like RBFOX3, we can identify the clusters that correspond to neurons. And using a general marker for astrocytes, such as SOX9, we can identify the clusters that correspond to those, and so on and so forth for oligodendrocytes and other glial cell types. But the cells that we were most interested in focusing on, of course, at this point was the neurons. And here's just, again, focusing in on that group where we can identify different groups that we collected during the dissection using more cell type specific diagnostic markers such as TRPC4 to identify the clusters that correspond to lateral septal neurons, and LHX6, we can identify those medial septal neurons. And then finally, to take advantage of the approach that we took, we can actually go back and then ask, based on whether those cells were positive for GFP or negative, of what is the contribution of NKX 2.1 derived cells to the diversity of these different populations. And things actually came out in really interesting ways. Some, more expected, such as that medial septal neurons, as I showed you, derive from a lot of NKX 2.1 expressing progenitors. The lateral septum has that mix, as we would expect, of both NKX derived and non-NKX derived populations. But one really interesting observation that we had was for the astrocytes, where hopefully you can appreciate these kind of two separate clusters of both non-NKX derived and NKX derived astrocytes that separate into these distinctive clusters shown by the colors. And when we look at the some of the genes that are enriched in those distinctive clusters and look at where those astrocytes that express those genes are located within the septum, we find that actually there's a pattern in which astrocytes, for instance, that are derived from the NKX 2.1 progenitors are nearly exclusively allocated into the medial septum. So they're spatially segregated from those cells that are enriched for, say, non-NKX expressing markers that I'll show you. So this is just an example using these astrocyte-rich markers that we found for glycine transporter and GABA transporter, and co-labeled them with this ALDH101, which is a pan-astrocytic marker. And we can do the complementary experiment and look at genes that were enriched in the clusters that correspond to those non-NKX derived astrocytes, or Zik derived, primarily Zik derived astrocytes. And you can see that there's this complementary pattern where in this case, we're using a glutamate transporter that's enriched in the non-NKX-derived cluster. And nearly all of those astrocytes are allocated to more lateral parts of the septum. So, we, again, we're most interested in this cluster of neurons that correspond to the lateral septum, partly because it's where there's the most interesting patterns of diversity that you can see. And we actually chose to subcluster that population. So we just took out the nuclei that correspond to the lateral septal neurons and subclustered them to get a kind of a better resolution view of the molecular diversity of just that subgroup of neurons. And here is just a tool that we use where we can both resolve the stability of the subclusters. But so this is a cluster tree where we're basically just looking at the patterns of clustering at different UMAP resolutions. And hopefully what you can appreciate is that the clusters that correspond to these different groups actually stabilize at relatively low resolutions. And this has also been helpful for us to look at the relationship of these different cells, so how they move as you increase resolution. So which clusters actually break up into successive clusters at the higher resolution. And at the resolution that we're examining, we actually identified approximately 13 clusters that we think might correspond to distinctive molecular subgroups. But again, we can go back and ask, what is the relationship of this diversity to their developmental history of NKX 2.1 expression? So if we just look at a plot of the same subcluster and just segregate based on whether they were positive for SUN1 GFP from the NKX 2.1 expressing population or negative, you can already see that those distinctive molecular subgroups can segregate. And if we look at these populations and actually do differential expression, and ask, what genes are differentially expressed by this group of clusters that corresponds to the NKX derived or the non-NKX derived. We actually made a really interesting observation in that one, there's several genes that seem to be differentially expressed just across the axis of whether they're NKX derived versus non-NKX derived. And one of the genes that was most heavily enriched in the non-NKX derived population is this transcription factor called MICE2. And another interesting observation that we had just from looking at the gene expression, although this was somewhat expected, is that if you look at the adult cells, the transcription factor NKX 2.1 itself is actually not very highly expressed. So the only way that we know that these cells are coming from NKX 2.1 progenitors is from our prospective genetic labeling. And we've been able to validate this, these relationships using immunohistochemistry, where we can use our NKX 2.1 Cree mouse line crossed to a TD tomato reporter and an antibody for MICE2. And we can see that, again, the NKX 2.1 Cree derived cells and the MICE2 expressing cells within the septum are largely non-overlapping. So it suggests that the data that we're getting from our single nuclei sequencing isn't an artifact of the sorting and separating of the cells, but is actually really reflecting the molecular features of the cells in the mature septum, like we hope. And then we can look at other markers of these cells and how they're distinguished across their distinctive axes. So here we focus first on the NKX 2.1 derived lateral septal neuron subgroups. And we've made a few interesting observations. So one that we found particularly striking is that a lot of these clusters that identify these molecular subgroups are defined by interesting transcription factors and also interesting kind of functional or effector genes. So, for example, transcription factors like DATCH2, LHX2, SOX6 define a few of the interesting molecular subgroups. So again, trying to get at this question of whether these subgroups are actually reflective of distinctive subtypes of neurons, we've gone again and looked in situ, this time using the Allen Brain Atlas, at the expression pattern of some of these marker genes that we've identified. And again, we can see, for instance, that DATCH2, which is actually expressed by two clusters here, the CHAT and the LHX2 cluster, has this very distinctive pattern that actually corresponds to a lot of the NKX 2.1 derived cells that we see with our fate mapping, where you get these kind of nice crescent patterns. But from that group and from our sequencing, we predict that DATCH2 is expressed by this population, and that CHAT and LHX2 make up a subset of the DATCH2 expressing population. And we actually see that. So the CHAT in situ is not great from the Allen Institute, and you can see the really high expressing CHAT cells in the medial septum, but hopefully you can see that in this ventral part of the lateral septum, it kind of corresponds to the bottom of that crescent, that you have this CHAT expressing population. Oops. And then for LHX2, it appears to us that it makes up the more dorsal aspect of the DATCH2 expressing crescents. And we can do the same thing with the mostly, Zik only derived population. And again, a similar pattern comes up where many of these clusters can be defined based on the expression of transcription factors such as ZEB2, FOXP2, and PAX6. And also, again, a lot of interesting effector genes that can define these distinctive clusters, such as this dopamine receptor D3 and this CART-PT, which are, dopamine receptor, and CART-PT is this cocaine and amphetamine responsive transcript. So it suggests that, possibly some of the functions of these neurons might be related to motivation or have implications for addictive behavior. And again, looking at the distribution of these cells, their patterns are largely non-overlapping, which again suggests to us that we might be labeling distinctive, these molecular subgroups might truly be representative of distinctive neuronal cell subtypes. And again, we can check the logic of some of these subgroups that we've identified. So for instance, with FOXP2, which is a part of the non-NKX 2.1 derived population, which we showed previously, that that population expresses the transcription factor MICE2. And we find that nearly all of the FOXP2 cells co-express MICE2. So in this case, FOXP2 makes up a subset of that larger non-NKX derived MICE2 expressing population. And then also, again, the rules that we've been able to infer from the sequencing seem to be maintained, that FOXP2 and NKX 2.1 derived cells do not overlap in their expression. So I think so far what we've learned from the sequencing of the mature septum, but with an eye to the developmental history of these cells, is that we can already see how the developmental history, in this case based on the expression of NKX 2.1, defines both the molecular identities of both glial cells with the astrocytes that can be divided into two major subgroups based on their NKX 2.1 expression history. And in the lateral septum, where we think we've identified approximately 13 putative non-overlapping molecular subgroups of neurons, and that these cells can also be, again, divided based upon their NKX 2.1 expression history and their current status of the expression of the transcription factor MICE2. So as developmental biologists, one of our major interests is not just in defining the diversity of these cell types, but understanding the genetic programs that actually lead to these specializations of these different diverse cell types. So we've attempted to try to get at this question by, again, using a single cell sequencing approach, but this time collecting cells over developmental time. So we took this approach to start during embryonic development at the start of neurogenesis and collect cells from the primordial septum at embryonic days 11 through 17, but then also to collect cells during the early parts of postnatal development, during the specification of these cells and wiring, all the way to P30 in what we consider the mature septum. And in this, we used this time a technique called N-drops, where we sequence the cells, but again, use similar analytical approaches where we can use Heatmaps to look at the clustering relationship of these cells. And what we have outlined in the colors are actually the cells that correspond or express diagnostic markers for progenitors, such as radioglia, intermediate progenitors, and then the clusters that correspond to newborn neurons. And we've used Heatmaps to identify a lot of the molecular identities, but we've also recently begun to use this tool called Spring, which essentially generates k-nearest neighbor maps and orders the cells. And we found Spring particularly useful for being able to infer the trajectories of the cells as they progress through the lineage and acquire their mature cell identities. And here's just an example of the Spring where we've collected all the cells, and you can see them over developmental time. And hopefully you can appreciate that already, just looking at the k-nearest neighbors map, that the earliest cells that you would expect are the progenitors are at the base of the projection, and the most mature cells at P30 are at the tips. And we, again, chose to focus on the neuron specification. So we were able to subsample those cells that express markers of neuroprogenitors and newborn neurons and produce a neuronal Spring cluster or a trajectory. And just to give a orientation for how the Spring plot has kind of helped us infer trajectories, we can look at the expression of the enrichment of diagnostic marker genes for different neuroprogenitor types and different stages in lineage progression, and see where those cells are enriched in the Spring plot. So for instance, this marker for radioglia, you can see that they are enriched at the bottom of the Spring plot. And then as radioglia, which typically divide and give rise to intermediate progenitors, will progress further up the plot. And then finally, we can use markers like double cortin to identify those newborn neurons. So again, this has allowed us to kind of infer the molecular trajectory of these cells that we've collected in the septum. And we've been able to also use Spring to look at single cell trajectories during both early and later time points in neurogenesis, at these times where we know that these different subgroups of neurons are being produced. So if you recall, some of the early work showed that more early parts of neurogenesis give rise to more medial septal neurons, and later neurogenic time points give rise to more lateral septal cells. So in the spring, we can infer the trajectories at these individual time points, but also we can use this visualization to see distinctive spatial subgroups of progenitors. So for instance, with the ZIC4 and NKX 2.1, we can identify those NKX 2.1 only regions of both the POA, parts of the MGE, and then also the regions of the septum proper and septal eminence, which would co-express both NKX 2.1 and ZIC family transcription factors. And we can again use markers to identify the molecular specification of the distinctive cell subtypes. So for instance, we can identify newborn MGE and POA derived cells by the expression of LHX6, cholinergic neurons with GBX2 expression, and the broader lateral septal neurons with the expression of this factor DNER. And using those markers, we can actually then start to look at the differential expression at both developmental stage and lineage stage specific times. So what I'm showing here are heat maps of cells that we've identified as either radial glia, intermediate progenitors, or those newborn neurons, and looking at their differential expression patterns at E11, E14, and E17. And what this can tell us about is the genes that are enriched at specific developmental stages, such as E11, E14, E17, but also at specific stages in the lineage. And it's these genes that are enriched that might be the candidates for regulating specific aspects of the competence states or molecular specification programs for the neurons that are born at these distinctive time points. So here's just a few examples for a transcription factor that's enriched in E11 or early radial glia using HMGA. And you can see that there's an enrichment of HMGA expressing, co-expressing, nestin co-expressing radial glia in the embryonic septum. Another marker we've identified is cyclin D2, which has the opposite pattern in intermediate progenitors, where it's most highly enriched in late born or in later embryonic stage intermediate progenitors. And one of the factors that was surprising for us to find initially is this transcription factor PRDM16, which is really highly enriched in newborn neurons in later embryonic stage septum. And the reason that PRDM16 was a bit surprising to us is that it's one of the regions where PRDM16 is expressed only in neurons. So in the septum, I should say, is where we've only seen expression of PRDM16 in postmitotic cells. And that's reflected also in our single cell trajectories, where you can see that the initial pattern where we expect PRDM16 to be expressed, which is in radial glia, where it's really highly enriched. But then for our trajectory, it looks like it goes down in the intermediate progenitor stage and then comes back on in newborn neurons. And this is really in contrast to some of the previous work that we've done that I'll show you, where we find that PRDM16 in other parts of the developing forebrain is exclusive to radial glia. But here in the septum, we can see actually that PRDM16 is expressed in radial glia, and you can see other forebrain structures along the ventricular zone where those radial glia reside, continue at highly expressed PRDM16. But the difference is that in the septum, there's this really high expression of PRDM16 in this mantle zone that corresponds to the region in which the lateral septal neurons are developing. So what we see is that there's this dynamic expression pattern just as we saw in our single cell trajectories where PRM16 is very high in the ventricular zone radioglia. It then is downregulated in the subventricular zone that corresponds to those intermediate progenitors, and then comes back on at really high levels in these postmitotic neurons in the mantle zone of the septum. And it's, one of the reasons that this was a bit of a surprise to us is that we had kind of a previous history in studying PRDM16, and it's shown that PRDM16 in the cortex is really exclusively expressed in radioglia. And here's just an image from that paper of the cortical ventricular zone and subventricular zone. And I'm not showing the cortical plate, but there's no PRDM16 expression in that region. And we've had previously shown that PRDM16 in expression in these radioglia actually functions to regulate the activity of developmental enhancers that are important for the specification of late-born upper layer cortical neurons. So in the PRDM16 loss of function, there's a deficit in both the production and the migration of these upper layer neurons in the cortex. And we had also looked at another lineage for PRDM16 function in the MGE. And here's just, again, a paper actually from Miguel, where we used NKX 2.1 Cre, which also targets the forebrain interneurons that are born in the MGE to delete that ventricular zone PRDM16. And from those MGE-derived cells, what we found is that there's a loss of interneurons. There's about a 30% reduction of cortical interneurons throughout the forebrain. And that this reduction, unlike what we saw in the cortex, appears to impact all of the lineages that are produced from the MGE. So both parvalbumin and somatostatin subgroups, which are the two major MGE-derived subgroups, both of those populations are actually reduced by approximately 30%. But this is in contrast to what we've observed, since we now know that NKX 2.1 is expressed by a subset of those lateral septal neurons. Miguel actually took a look at the septum of those NKX 2.1 Cre conditional mice. And it was a pretty shocking finding to us at the time, which is that those lateral septal neurons that are derived from the septal limitants are completely ablated. However, the cells that are populating the medial septum appear to be entirely unaffected by the loss of PRDM16. And we've been able to, again, kind of go back and take advantage of our sequencing to look at other molecular markers of NKX 2.1 derived cells in our PRDM16 knockouts and show that those cells are gone. And so one, that the cells are gone, but then also that these molecular subgroups that we identified with our sequencing, that those cells can't be compensated for. So here we're showing CRHR2, which is expressed by the subgroup of NKX 2.1 derived neurons. You can see in the PRDM16 conditional knockout that we lose the expression of that CRHR2. Whereas if we look at markers that are enriched from non-NKX derived lineages, the representation and organization of those populations is completely unaffected. So, one of the questions we had is, we've looked across these different lineages, including the cortex, the MGE, and now in the septum. And it's why is it that these septal neurons are so uniquely susceptible to the loss of PRDM16? And I think, one of the clues that was right in front of us is that it is these septal neurons that are the only populations of cells that express PRDM16 postmitotically. So it's only these lateral septal neurons that continue to express PRDM16. So to really test whether it's this neuronal expression of PRDM16 that's necessary for the survival of these cells, a grad student in the lab, Chris Reed, did this experiment where he took advantage of a mouse genetic line, this DLX56 Cre, that's exclusively expressed in GABAergic neurons and not progenitors. So here's, again, just using the Sun1 GFP reporter. You can see the labeling within the septum, but Chris also went back and used a marker for progenitors to confirm that the recombination is taking place exclusively in the neurons. So you can see that there's not a lot of overlap with the Sun1 GFP and this Ki67 positive population of progenitors. So with that confirmed, he then crossed this DLX56 Cre into our PRDM16 conditional animals. So here's the control, where you can see this time using the TD tomato reporter that the septum is kind of normally organized. But then in the PRDM16 conditional knockout, we get this massive reduction in the lateral septal neurons. So it does indeed appear that it's PRDM16's neuronal expression that's necessary for the survival and development of neurons in the lateral septum. So just to summarize here, I told you in the beginning about work where we've been using a lot of single cell sequencing approaches to identify both the molecular diversity of mature neurons and the molecular programs that produce their diverse specification. And one of the factors that we've identified both in our sequencing and from our genetic approaches, PRDM16 appears to be required for the survival and potentially the specification of these neurons within the lateral septum. So we think that this NKX 2.1 Cre PRDM16 knockout actually creates kind of a cool opportunity to explore both the development of the circuit and its plasticity with the perturbation of this distinctive subset of neurons. And the future work that we're really, really excited about is, we now have molecular handles on a lot of these distinctive subgroups, and we can start to ask, what might be some of the key functional specializations of these diverse groups of lateral septal neurons. So what might be their unique circuits, and of course, what are some of the behaviors that are mediated by these distinctive populations? And then, of course, we have all of our favorite developmental questions, such as what are the lineage relationships between these cells, especially those cells that are derived from, say, the septal eminence that produce cholinergic neurons, the subset of lateral septal neurons, and so on. And there's also a lot of really interesting questions with this unique neuronal expression of PRDM16 is, what are the potential unique molecular functions of PRDM16 in these septal neurons? So just to acknowledge the people who have actually done the work, Miguel Turo Garcia is the postdoc in the lab who actually made some of the really initial key discoveries initially from the PRDM16 knockout that actually put us on the track to beginning to study the septum. And Chris Reed is a graduate student in the lab who's really kind of jumped into a lot of the questions related to the molecular diversity of those mature septal cell types and characterizing some of the functional properties of those cells. And just a lot of our collaborators for a lot of the work. So from Mike Greenberg's lab, Sanisha and Harwell, who helped us with a lot of the end-drop sequencing, Caleb Weinreb from Alon Klein's lab, who was really critical in helping us get the spring going and starting to do some of that initial analysis. And then, a lot of this work was funded through my time at Harvard through the Harvard Brain Science Initiative, the Giovanni Armanese Harvard Foundation, and of course, our funding support from the NIH. So I'm gonna stop sharing here, and I don't know how much time we're looking like on time, but
we'll do about 6 or 7 minutes for Q&A. So thank you so much, Corey, for that incredible talk. If anyone has questions, I see a couple have come in through Zoom's Q&A function, but if you have additional questions for Corey, feel free to drop them there and I'll ask them directly. For our first question, it comes from Joe Yu. They asked, out of curiosity, have you run any trajectory analysis on the single nuclei data? If so, how well do they perform in terms of inferring origin and branching points for the different cell types? And then the second question is, do the predicted trajectories match up with the data from your functional validation experiments?
Yeah, so those are kind of experiments that are now underway with some of the nuclei data. So we haven't done trajectories with the single nuke data just because it's mostly the largest data sets that we currently have are from the mature. So it's really we've been focused on the questions of looking at the molecular identities of those distinctive groups rather than questions related to their specification. But we are actually really excited to do additional nuclei, single nuke experiments at these developmental time points, which we think might give us better resolution to use some of the other trajectory tools to kind of infer the molecular programs for specification of these neurons.
Wonderful, thank you. Our next question comes from Oliviera Junior Markley. They said, congratulations, Corey, amazing talk. They also have 2 questions. One, is there anything in particular at the medial wall of the SVZ that holds astrocytes that have been born there to stay at the lateral septum? And 2, have you all looked at the extracellular matrix organization and configuration within those 2 areas of the septum? And how about maybe 3 questions? How about the gene profile of metallopeptidase inhibitors of septum medial SVZ astrocytes? So I think you can take them all if you want to like break them all down.
Yeah, so yeah, we actually don't know. So we don't know. It's another area that we've over the past few years have become really interested in glial cells as well. And I think there's a lot of really cool yet to be addressed questions related to glial development. And I think that the septum provides a really interesting platform for asking those questions because we have this now developmental perspective on the origins of the cells and how early on do these molecular programs position those cells and what aspects of their molecular identities are intrinsic and related to their developmental origin versus the environments that they migrate into that also shape their mature function. So we don't know about anything in particular yet, but we think again that these data sets, we're excited that we might find some interesting clues that can give us a handle on some of those questions. And kind of the same thing for the ECM. So there's definitely interesting expression patterns in both the developing and mature septum of different ECM components that we can see, across both the mature and the developing septum. But what's the function of any of these particular factors, either in the mature septum or during development? We still we don't know.
Great. Thank you. Okay, I have a question. This is less about the science, more about your career trajectory. So as someone who is trying to wrap up a postdoc and hopes to start a lab, what is some advice that you wish you had as you were making that career transition in terms of navigating, starting up your lab for the first time, and particularly what might be useful for someone as a person of color?
Yeah, I think that a lot of my successes have happened because I've had a lot of key mentorship and support at really critical stages in my career development. And I think being able to find those advocates and along the way is really important. I think that's important for when you're looking at places to evaluate whether that kind of advocacy and mentorship is a component of the culture in the environments that you're going into, because there's a lot of things that I think as a postdoc, you know about the science, but there's many aspects to being a PI, and especially being part scientist, part administrator, and part manager, and so on, that it's not intuitive how to do all of these things. So I think that it is important to have people to, and I think also for us, so again, thinking about Black people especially in the United States coming into science, there's a lot of things I think in science where there's for a long time been a lot of kind of unspoken rules or unspoken things that are not made explicit, but somehow everybody kind of implicitly knows that that's how you're supposed to kind of conduct yourself. So I think finding people that can help you navigate those things, people who will read your grants and give you honest feedback, and that takes a lot of time. So I think really being able to find a couple of those advocates as you're starting to build up your research program is really going to be critical.
Thank you so much. Thank you for that answer. I know that's a thing that especially people in my position, but even earlier as a graduate student, when you're thinking about your career trajectory, just like good things to keep in mind for the future. And a lot of us don't have access to people like you who can answer those types of questions. So that's so valuable. And you just being here and being as successful as you are, I'm sure you'll be an inspiration to many people listening, as you were for me when I was kind of just getting started. So I want to
appreciate that.
Yeah, I want to, I guess we'll close because we're at time, but I just want to thank you again for such an incredible talk. Thank you for taking the time when I know your time is so limited, having just moved your lab across the country in a whole pandemic. I want to remind people of some upcoming events. We have a social during SFN next week, a virtual social, and that's going to be next Thursday, November 11th, from 2:30 to 3:30 PT, 5:30 to 6:30 ET. Also, the last of our seminar, and Angeline just dropped the link to register for the social in the chat. And then our last seminar series of the year is going to be a month from now, the first Friday in December, the first Friday in December. It's going to be with postdoc Austin Coley at the Salk Institute. So definitely stay tuned for that. As we wrap up, I wanted to thank our president and Wonder Woman, Angeline Jukes, for providing technical support today and just all of the other amazing things that she does. The other co-organizers on our seminar series organizing committee, Ashley Cunningham and Paige Greenwood, the rest of the BIN team. And then I want to thank you all for being here with us and sharing space today. And I hope that you got something valuable from it, because I certainly did. So thank you so much and have a wonderful Friday.